Hi everyone, this is lecture 20. I am back doing the videos for the foreseeable future. This section is on cell division, specifically mitosis and meiosis, and material is found in chapter 12 in your textbook. A lot of this might be review, but you know what? Review is a good thing. Let's start with some questions. Number 1, which of the following occurs in mitosis but not in meiosis? Number 2, homologous chromosomes. And number 3, without crossing over, an organism with 3 pairs of chromosomes can produce how many possible different gametes? All right, let's begin. So, up till now we've talked a lot about the molecular genetics. How does DNA get transcribed into RNA, translated into protein? How do mutations occur? How does that lead to a phenotype in terms of protein expression? What we're going to be turning our attention to now in the rest of the semester is inheritance. How do these mutations get passed on? What patterns of inheritance are there, for example, recessive dominant? We're going to be talking about cell division because it's so critical for understanding that process. Let me tell you, first of all, about the cell cycle. Shown here in sort of this crazy diagram is a cell cycle depiction where we have 4 phases. G1, which is the growth phase, number 1. S phase, which is synthesis, DNA replication occurs here. G2 is a secondary growth phase. The cell is preparing itself to divide. And finally, highlighted in blue here, is mitosis, cell division. You can see that it's only a small portion of the entire lifetime of a cell. Also shown on the slide is a representative DNA molecule, shown here with 2 strands, double helix, black, one strand black, one strand blue, connected by a centromere. In humans, we have 46 of these, 1 DNA double helix per chromosome. And this is what the DNA will look like in G1 phase. As we saw at the beginning of the semester, the DNA replicates. This process occurs during S phase of the cell cycle, DNA synthesis. The DNA that's replicated is replicated semi-conservatively and produces 2 new daughter strands, which are shown here, black and blue. And these are attached at the centromere, as shown here in eukaryotes. So after S phase, our cells contain 46 replicated chromosomes. Or another way to think about that is that each arm of the chromosome is called a chromatid. There are 92 of these because they have replicated, they have doubled. 46 times 2 equals 92. After S phase is complete, the cell prepares itself for entering mitosis. The chromosomes will begin to condense through a process of heterochromatization. That picture is shown here. 46 condensed chromosomes. Mitosis, cell division, will occur next. The chromatids separate. As you can see, one half goes into one daughter cell, the other half go into the other daughter cell, thereby giving you 2 daughter cells that have an equal number of chromosomes. So let's talk a little bit more about cell division. I'm going to give you a couple examples of when cell division is super important and one example of a case when cell division goes haywire. First off, cell division is super important for development. You take an unfertilized egg, it fertilizes, and then begins dividing and dividing and dividing to create an entire organism. This process of division involves the process known as mitosis, cell division. Like we just showed in the previous slide, every time the cell divides, it must replicate its DNA and then divide that DNA equally between the 2 daughter cells. Eukaryotic cells divide to produce more cells and increase the size of the organism. Adult humans have over 4 trillion cells. That's not counting all the red blood cells and white blood cells we have. These are all produced by division from one starting egg and sperm. Pretty amazing. In adults, however, cell division is also very important. And this is one example where it's thought that the primary way that our cells go through cell division is from a stem cell, an adult stem cell. For example, it could be a hematopoietic stem cell, one that produces blood. These stem cells divide by mitosis, equal DNA going in each of the two daughter cells. One daughter cell will continue to differentiate into the final mature cell, whereas the second daughter cell will remain a stem cell. It's called self-renewal, and will able to produce more differentiated cells. This is how most of our organs are maintained: epithelial cells, blood cells, brain cells, and so on. Sometimes cell division can go haywire, as in forming tumors, and in this case metastatic tumors called cancer. Here is a picture of a normal epithelium showing individual cells lying on a layer of basal lamina. If cell division has a mutation, if the genes have a mutation that allow the cells to divide uncontrollably, they form a tumor in situ, meaning in place. Additional mutations might accumulate, however, which allow the cells to grow and metastasize or spread throughout the body through the blood or lymph system. In that case, they can travel to other sites, take up residence, and begin replicating there and dividing, forming additional tumors. This is what we call metastatic cancer. Alright, so mitosis is really important for all those processes. One cell divides to give rise to two genetically identical daughter cells. Equal DNA goes to both daughter cells. Same number of chromosomes as the parent cell. Remember, the DNA replicates so that you double up the amount of DNA, then you divide it evenly between the two daughter cells. The arms of the chromosomes are called chromatids. We call these sister chromatids. They're attached at the centromere, and they separate prior to cell division. This is a schematic from your textbook showing the steps of mitosis in more detail. It starts here at the top. This is a cell that's in what we call early prophase. Prophase, you'll notice that the nuclear membrane is still intact. The chromosomes are not yet condensed but are beginning to condense. Through the process of prophase, the mitotic spindle begins to form. The chromosomes condense, and here this cell has 4 chromosomes, 1, 2, 3, 4. And there are 2 sets of homologous chromosomes. The blues are homologous to each other, and the orange chromosomes are homologous to each other. In metaphase, the next stage, the chromosomes becoming attached to the spindle at the centromere. And the centromeres and the spindles push those chromosomes into the center of the cell. They line up into metaphase in no particular order. It's completely random. In anaphase, the next stage, the centromeres that are attached to, the spindles will begin pulling at the centromeres and pulling the chromatids apart, pulling them to the opposite poles of the cell. This continues on in late anaphase and into telophase. Telophase is where the nuclear membrane begins reforming. The spindle begins to break down. Then the cell continues back out of mitosis and into G1 phase, so-called interphase. Interphase just simply refers to any other stage of the cell cycle that's not mitosis. The nuclear membrane is completed now. The cells are a little bit smaller. They need to grow again. And this DNA has decondensed into its normal state. So that's mitosis. Again, that's very important for development, for maintenance of adult tissues, and it goes haywire in diseases like cancer. One quick note about the spindles. These spindles connect to the centromeres, which are the regions of the chromosome that are kind of constricted. These centromeres can bind to proteins which are collectively known as a kinetochore. Kinetochore complexes bind to the spindle fibers. They are the ones that physically will then pull the chromatids apart. Okay, that was mitosis. We're going to be speaking mainly about meiosis, though, going forward. And the reason why is because meiosis is very important for understanding inheritance and how do genes and traits get passed along to the next generation. Meiosis is very similar to mitosis, except that it has 2 cell divisions following only 1 DNA replication. Meiosis particularly produces gametes. Gametes are your eggs and sperm, and they have half the number of chromosomes that all of your other cells have. We call these cells haploid cells because they only contain half of the normal DNA complement. That's opposed to your normal cells, your somatic cells, which contain a diploid DNA content, or 2 of each chromosome. This slide depicts mitosis and meiosis side by side, showing the similarities and differences. The big difference at the beginning in meiosis is shown right here in the middle. Here, the homologous chromosomes are shown as red and blue. Notice that they are pairing up in metaphase. This is the meiotic division 1. The meiotic division 1 metaphase, sometimes called metaphase 1, features pairing up of the homologous chromosomes. This is in contrast to mitosis, where the homologous chromosomes do not pair up. They randomly assort during metaphase. Then, during the first cell division, the homologous chromosomes will be pulled apart by the spindle fibers, so that each daughter cell in meiosis division 2 will contain only 1 of the 2 homologs. That's division number 1. Division number 2 then is very similar to mitosis in the sense that the spindles pull the chromatids apart, giving you, finally, individual gametes, shown here with the yellow, that contain a single DNA molecule of each type of chromosome. So 2 separate cell divisions following only 1 DNA replication. On this slide, I'm introducing the symbol N. N refers to the number of chromosomes in a haploid gamete. Haploid cells, for example these ones at the bottom here in yellow, contain 1 of each chromosome type. For example, chromosome 1, 1 chromosome 2, 1 chromosome 3, etc. In humans, our gametes contain 23 chromosomes, 23 individual chromosomes. In most diploid cells, however, in diploid cells, in our somatic cells, our DNA content is 2N. 2N refers to the number of chromosomes in the diploid genome. We have 2 chromosome 1s, 2 chromosome 2s, 2 chromosome 3s, etc. In meiosis, a cell that has a diploid genome, 2N, will divide in the first cell division, producing daughter cells that have only 1N, 1 of the 2 homologous chromosomes. Contrast this with mitosis, where the cells remain diploid the entire time. Because both daughter cells end up with 2 copies, as shown here at the bottom in yellow, they remain diploid the entire time. Okay, that is all for today. I will see you guys in class.